When an organism reproduces, its genetic instructions are passed on from one generation to the next through the process of DNA replication. Now, because DNA has the amazing ability to serve as a template, it can create an exact replica of itself, thereby creating two identical strands of DNA. First, we have the enzyme helicase, which unwinds the original DNA molecule into two separate strands. Single-stranded binding proteins keep the strands from coming back together. Two strands begin to form, the leading strand and the lagging strand. The leading strand is synthesized non-stop in the 5-3 direction by DNA polymerase. The lagging strand is synthesized discontinuously. Primase synthesizes a short RNA primer. DNA polymerase later extends these DNA primers to form an Okazaki fragment. After the RNA primer is replaced by another DNA polymerase, DNA ligase joins the Okazaki fragments to the growing strand. Replication continues on both strands until there are two exact copies of DNA molecules.
The flow of genetic information in a eukaryotic cell happens when a sequence of nucleotides in the DNA is transcribed into a molecule of RNA in the cell's nucleus. The RNA travels to the cytoplasm, where it is translated into the specific amino acid sequence of a protein. Here's another way of describing the flow of information in a eukaryotic cell. When the building designer lays out the blueprints for a new building complex, he first makes a copy of the blueprints. These blueprints are then sent to the builders, which then translate the designer's blueprints to the ground on which the buildings will be made.